Hello everyone, and welcome back to your online video lectures. This chapter is all about the four major macromolecules: carbohydrates, lipids, proteins, and nucleic acids. We'll talk about the monomer units that make up the polymers and really dive into the structure and functions of these crucial organic molecules. In this chapter, we will continue to model and categorize the fundamentals of molecular structure and functions associated with living organisms. Specifically, you will be able to identify and describe the structure-function relationship of carbohydrates, lipids, proteins, and nucleic acids. All right, this chapter has a lot of information, but this is one of those chapters that will serve you in every single future science course. If you're interested in biochem, nutrition, endocrinology, physiology, or literally any other science course, do your best to learn this chapter. All future health science courses will make much more sense to you. And as a bonus, this chapter will arm you with the knowledge to help make more informed health choices in food, beverage, and supplementation. For this chapter, in addition to your typical note-taking, I recommend that you track information in a table like this. And I recommend filling it out as we move through this video, so pause and fill in as you go. As I've mentioned, all living things are made of four classes of large biological molecules: carbohydrates, lipids, proteins, and nucleic acids. These large biological molecules are called macromolecules. Let's break down that word. No doubt you are familiar with micro. Micro comes from the Greek word mikros, meaning very small. We need a microscope to see microbes, which are very small, single-celled organisms. Macro comes from the Greek word makros, meaning long or large. We can take a macroscopic look at a situation to see the larger picture. We learned in the first chapter of emergent properties, which are properties that manifest themselves as the result of various system components working together. Each of the large biological molecules that we're about to learn about have unique emergent properties that arise from the arrangement of their atoms. To understand macromolecules, we have to understand what they're made of. Macromolecules are polymers, which are built from monomers. Monomers are repeating units that serve as building blocks, and polymers are large molecules consisting of many of these smaller building blocks. Mono means one, while poly means many. In this case, the polymers are the macromolecules, carbohydrates, proteins, and nucleic acids. And just note that lipids, the fourth macromolecule, is not a true polymer because they're not made of repeating monomer units. We'll see that when we get to lipids. So, how do we build these polymers from monomers? Here is your intro to that special group of molecules called enzymes. I'm sure you've heard of enzymes before. Enzymes are specialized macromolecules that speed up chemical reactions, such as the ones that are making or breaking polymers. Enzymes are absolutely crucial to life. Every single organism has enzymes to help speed up the metabolic reactions that are required for life. We will learn more about enzymes in Chapter 8. One kind of reaction that enzymes help to catalyze is called a dehydration reaction, and this is also sometimes called a condensation reaction. So don't get confused if you see both used. A dehydration reaction occurs when two monomers bind together through the loss of a water molecule. So let's look at that. In image A, we're shown a polymer consisting of three purple monomers. At the ends of this polymer are an OH hydroxyl group and an H atom, hydrogen. This is common for the macromolecules that we're going to be learning about. We are also shown a single purple monomer with an OH hydroxyl group on the left and a hydrogen on the right. In order to bind that single monomer to that polymer, we need to remove the hydrogen and the hydroxyl OH, highlighted in blue. Together, that H and OH will form that water molecule H2O. This is the dehydration reaction, the loss of water molecule when monomers bind together. And you know what the word dehydration means, so that's what's happening here. We're losing water. The result is a polymer consisting of 4 monomers and the release of that water H2O molecule. And remember, this reaction is catalyzed by an enzyme. This is how we assemble polymers. So how do we disassemble polymers? We use a reaction called hydrolysis. Hydrolysis is essentially the reverse of that dehydration reaction. In image B, we're shown that 4 monomer polymer that we just created using the dehydration reaction. In order to remove a single monomer from that polymer chain, we need to add a water molecule. That H2O is split and placed onto the ends of that polymer and monomer. Just as an enzyme was used in the dehydration reaction, an enzyme is used here as well. As you progress through your courses, you'll see these enzymatic reactions, dehydration reaction, and hydrolysis are used to build many different polymers of the body. In the last chapter, we learned about carbon and its role in creating large, complex molecules. We learned that molecules containing carbon are called organic molecules, which are central to living organisms. We also learned that carbon is the backbone of most complex organic molecules, notably the 4 macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Carbon's versatility and the ability to create 4 covalent bonds lends to that great diversity in polymers that we see in living organisms. Cells have thousands of different macromolecules, which can vary among cells of a single organism. They can vary within a single species, and they can vary between different species. That versatility of carbon lends to this huge variety of polymers that we can build from a small set of monomers. So let's get into those macromolecules. Macromolecule number 1 is the carbohydrates. In the body, carbohydrates serve as the fuel and building material of our cells. Carbohydrates are the sugars and polymers of sugars. I know when you hear the word sugar, you probably think of table sugar or a number of different artificial sweeteners that you can use. But from now on, I want you to redefine what a sugar is. Table sugar is specifically the molecule sucrose, which is just one type of carbohydrate, which is one type of sugar. All carbohydrates are considered sugars. This ranges from the carbohydrates that make up breads and pastas to the sweet sugars in fruits called fructose or milk called lactose. Carbohydrates are also referred to as carbs, so I'm sure you've heard of that before. And we have a couple of terms to learn for carbohydrates. Monosaccharides are simple sugars. They are the monomers for the carbohydrate macromolecule. Polysaccharides are the macromolecules. They're the polymers of carbohydrates. Let's look at the monosaccharides first. Remember, monosaccharides are the simple sugars, the repeating units for carbohydrates. These repeating units are usually in multiples of CH2O. We'll look at that in just a second. Glucose is the perfect example of a monosaccharide. Glucose is the most abundant monosaccharide in the body. It circulates in the blood of animals as blood sugar and is the most important source of energy for all organisms. When we learn about photosynthesis and cellular respiration in later chapters, glucose will be at the forefront of those discussions. Glucose has the molecular formula of C6H12O6. We see glucose in the bottom left of the table of sugars provided. Each of the molecules shown in this table are examples of monosaccharides, simple sugar monomers. Monosaccharides are classified by two different things. Number one, the location of the carbonyl group as an aldose or a ketose. For a refresher on the functional groups, see the Chapter 4 video that is posted. A carbonyl is an oxygen double bonded to a carbon. In the table shown, all of the carbonyls are highlighted in orange for easy viewing for you. Take a moment to see where the carbonyls are located. Some are at the top of the alpha carbon, and others are attached to centrally located carbons within the molecule. On the top of the table, we see the words aldoses and ketoses. Molecules that have that carbonyl at the end of the molecule, where the carbonyl carbon is flanked by a carbon and a hydrogen, that is an aldehyde group, so those molecules are called aldoses. Remember, these are aldehyde functional groups. Molecules that have the carbonyl central within the atom, where the carbonyl carbon is flanked by two carbons, are called ketoses. And remember, these are ketone functional groups here. Number two, the second thing, is the number of carbons that are present in the carbon skeleton. This table is further divided by the number of carbons present in the molecules. The top two molecules both contain 3 carbons in the skeleton chain. These are called trioses, right? Tri means 3. The middle 2 molecules contain 5 carbons, and these are called pentoses. And the bottom 3 molecules contain 6 carbons. These are called hexoses, right? So we have tri, pent, and hex. Notice all of the hexoses shown have the molecular formula C6H12O6, even though they are all different molecules. Do you remember what this is called from the previous chapter? These are all examples of a type of isomer called a structural isomer. And it's the same for the pentoses and trioses shown as well. The last observation for this table that I want you to look at, what do these molecules tend to end in? All of the molecules that I just talked about. They all end in ose, O-S-E. Sugars, carbohydrates, carry the suffix ose to denote that they are carbohydrates. Examples are glucose, fructose, lactose, maltose, sucrose, and ribose. Let's take a closer look at glucose. The linear version of glucose at the top left was shown to you in the previous slide. Although glucose is often drawn in a linear skeletal structure like this, glucose doesn't actually exist linear in your body. In an aqueous solution, which is one where water is the solvent, such as our bodies, right, our blood, our tissues, our cells, glucose and many other sugars form a ring. The chemical reaction on the top shows how glucose changes from that linear structure into the ring. get used to seeing monosaccharides in their ring formation. They're usually shown in pictures as pentagons, little 5-pronged circles, or a hexagon with 6 points on it. So let's start using our monosaccharide rings to build some larger carbohydrate polymers. First, we'll build a disaccharide. A disaccharide is formed when a dehydration reaction joins two monosaccharides, right? Di means two. As we move through how to build all of the different polymers, I'm going to point out the names of the bonds that are used to create them. Make sure you note them and write them in that table that I provided in the beginning. When we join two monosaccharides together using that dehydration reaction, a covalent bond called a glycosidic linkage is formed. Remember, carbon makes 4 covalent bonds. All of the bonds that hold the monomers are going to be covalent. Your carbohydrates, your lipids, your proteins, and your nucleic acids are all held together by covalent bonds. Image A shows this dehydration reaction. When you join two glucoses together with that glycosidic linkage, you synthesize the disaccharide called maltose. In image B shows another dehydration reaction. This time we're joining glucose and fructose together with a glycosidic linkage, and we are synthesizing sucrose, which is table sugar. Disaccharides are the smallest polysaccharide, right? 2 is the smallest multiple that we can have. Our body can make much larger polysaccharides, which are used for storage and structure in our cells. 3 examples are starch, glycogen, and cellulose. You probably have heard of those 3 before. Starch is the storage polysaccharide for plants, and it's consisting of glucose monomers. You may know of starch from potatoes. In image A, we see the structure of starch. Notice those glucose monomers are shown as green hexagons. Each one of those is a glucose monomer. The glucose monomers are either attached in a linear formation or they're branched. The simplest form of starch is called amylose. The glucose monomers are joined in just a linear chain, and if we add branches to amylose, we create another type of molecule called amylopectin. As a reminder, all of the monomers on this page are joined again by glycosidic linkages, bonds that join sugar monomers to form carbohydrates. Next is glycogen, which is shown in image B. Glycogen is the storage polysaccharide for animals like us. As you can see, glycogen is also a polymer of glucose monomers. We see glycogen has both linear chains and is branched as well. When you eat a meal that is rich in glucose, your body has 2 options. It either must use that glucose as energy to fuel your metabolism for movement or whatever it is that you're doing, or it has to store it. It stores that excess glucose for later use. Our bodies are experts in storing excess energy. The first way that it will store excess glucose is in this new structure called glycogen. The primary glycogen storage sites in our body are our liver, which has about 100 grams, and actually in our muscles, which altogether store about 400 grams of glycogen. And why is glycogen so important? Remember, our body, and particularly our brains, require a certain amount of glucose to be circulating in our blood at all times. This amount is about 90 milligrams per deciliter. That's a normal fasting blood glucose level for us. We have a mechanism called negative feedback, right? Which ensures that this level of blood sugar is maintained at all times. So when we go through periods of starvation, for example, while you sleep at night and you're not taking in any excess glucose or energy, glycogen becomes the body's source for glucose. Altogether, the average person holds about 1,500 to 2,000 calories worth of stored glycogen. This is about 24 hours worth of stored energy for you just in your glycogen. Recall that hydrolysis is the process of breaking off a monomer from a polymer. When our bodies need energy, for example, while we're sleeping at night, hydrolysis will remove a glucose monomer from the glycogen chain to be released into our bloodstream so that we can use it to make energy while we sleep. The last polymer is cellulose, another molecule that is found in plants. Cellulose is the major component of the tough cell walls of plants. I'm sure you all remember that, the difference between animal cells and plant cells. Plant cells have that tough, sturdy cell wall, and it's made of cellulose. We can see that cellulose is also, again, another polymer of glucose. Glucose is the monosaccharide here used. However, the glycosidic linkages here are different. Notice the two differing ring formations of glucose at the top of this slide in image A. On the left, we have an alpha glucose, and on the right, we have a beta glucose. The difference is which direction that hydroxyl OH on carbon 1 is oriented. It's down on alpha glucose, and it is up on beta glucose. Now look at image B for starch. The 4 glucose monomers are all in the alpha glucose orientation. That hydroxyl group was down prior to binding. This type of glycosidic linkage is called a 1,4 alpha linkage. Let's take a quick detour to learn how to count carbons in a sugar ring. It will come in handy later on when we get into DNA. So to count carbons, you start at the oxygen that's present in that ring, and you go clockwise around the ring. The first carbon is carbon 1, then carbon 2, 3, 4, 5, and 6 is over here. The 1,4 linkage is called that because that bond, that glycosidic bond, is between the carbon 1 of the first monomer and the carbon 4 of the second monomer. Now look at the cellulose in image C. This is a 1,4 beta linkage. The monomers are beta glucose monomers with every other monomer inverted. Again, the glycosidic bond is between carbon 1 and carbon 4 of these molecules. When we consume cellulose, meaning when we eat plants, it passes through our digestive tract undigested, and we call this, you've probably heard this before, fiber, specifically insoluble fiber, because we're not able to digest it properly. And this is because we lack that enzyme, a specific enzyme that's required to break the 1,4 beta linkage in cellulose polymers. Herbivores like cows also, fun fact, lack this digestive enzyme to break up cellulose, which is Again, cellulose is abundant in the grasses and the feeds that cows eat. However, cows have developed a symbiotic relationship with microbes that are present in their gut, in their digestive tract, which do carry the necessary enzymes. Therefore, cows are able to derive energy from cellulose, unlike humans. The final example of a carbohydrate polymer is called chitin. Chitin is a structural polysaccharide found in the hard exoskeletons of arthropods. The monomer of chitin is pictured here. Just notice that it is a modified version of a glucose monomer. I don't think you would need to know anything beyond that for this class. And now we're on macromolecule number 2, which is lipids. Lipids are a diverse group of hydrophobic molecules that do not actually have true polymers. The one thing that all lipids have in common is that they don't mix well with water. They consist mostly of hydrocarbons, hydrocarbon regions. We learned about hydrocarbons in the previous chapter. Hydrocarbons are molecules made solely of hydrogen and carbon. And be careful not to mistake hydrocarbon with carbohydrate, because they're very different. Remember, with hydrocarbons, since those bonds are between a hydrogen and a carbon, those two atoms have very similar electronegativities. So that covalent bond will be nonpolar. And this is why lipids are hydrophobic. They're mostly nonpolar. The 3 most important lipids are fats, phospholipids, and steroids. So lipid number 1 is fats. And I know you've heard of fats, but now we're going to actually see how fats are constructed, what they actually are. So fats are made of 2 types of smaller molecules, glycerol and fatty acids. Glycerol is seen here highlighted in gray. It's a 3-carbon alcohol, and notice how it ends in the suffix OL, which denotes that it is an alcohol, such as ethanol. Each carbon contains a hydroxyl group coming off of it, and this is where the fatty acid chains are going to connect. The fatty acid chains are highlighted in yellow, and notice that they're all hydrocarbons, meaning they're just made of hydrogen and carbon, and that they vary in length. Fatty acids consist of a carboxyl group attached to a long carbon skeleton. The carboxyl group is where the attachment occurs, and it hooks onto the hydroxyl groups of the glycerol, and this forms a new linkage called an ester linkage. So note that for lipids. And what we've created is a molecule called a triacylglycerol, or it's also called a triglyceride. As I mentioned, the one thing that all lipids have in common is that they do not mix well with water. Fats separate from water because water molecules hydrogen bond with each other, and in doing so, they simply exclude the fat molecules. So let's look into those fatty acids a little bit more. The fatty acid chains can vary in length and in the number of double bonds. I'm sure you've heard of the term saturated and unsaturated fats, and now you'll learn what that means. When the hydrocarbon chain, the fatty acid chain, has no double bonds, this fatty acid is called saturated. When that fatty acid chain has double bonds, it's called unsaturated. That's simple. Unsaturated fatty acids can have 1 or more double bonds. You might have heard of monounsaturated fatty acid, which would just have 1, or polyunsaturated fatty acid, which would have more than one. Saturated fats with no double bonds tend to come from animal products. For example, butter and the marbling that you see in animal meat. And they tend to be solid at room temperature, and this is due to that linear structure that those saturated fatty acids have. You can see the difference between the saturated and unsaturated. The saturated fats are linear, which allows them to pack in tightly together, making them less fluid. Unsaturated fatty acids come from fish fats and plant fats. For example, we have olive oil and fish oil and sesame seed oil, and those all tend to be liquid at room temperature, which is due to the kinked structure of the unsaturated fatty acid. So those don't pack in as closely together. Remember, when double bonds are introduced into a hydrocarbon, the molecules become planar and are unable to rotate freely around the double bond. The saturated fats found in animal products, such as butter, are believed to contribute to cardiovascular disease due to the formation of plaques. Again, this is in part due to that linear structure that these saturated fats take on. These fatty acids can pack in much more tightly together than those kinked unsaturated fatty acids. You may have also heard of hydrogenated oils. Hydrogenation is the process of converting unsaturated fats to saturated fats, and we do this by adding hydrogens. This converts the double bonds into single bonds. And you've probably also heard of trans fats. So recall from the last chapter, the type of isomer called a cis-trans isomer. And this has to do with where the functional groups fall around a double bond. Trans fats are unsaturated fats with the molecular groups on opposite sides of the double bond. Trans fats are said to contribute to cardiovascular disease even more than saturated fats are. Our bodies use fats as a mode of energy storage. We have these specialized cells called adipocytes or adipose cells that store lipid droplets as a source of long-term food reserves. Adipose tissue also serves a vital role as cushion for our organs and insulates to help keep us warm. The second type of lipid is a phospholipid. Phospholipids are the building blocks of cell membranes. The structure of the phospholipid is very important in understanding how it functions in cell membranes. Take a look at this image. In the phospholipid, two fatty acids and a phosphate group are attached to a glycerol. The two yellow fatty acid chains, which we call tails, are hydrophobic, which means that they're water-fearing, and this is because they're hydrocarbons. Notice in this phospholipid, we have one linear saturated fatty acid and we have one kinked unsaturated fatty acid. This can change. Your body can make whatever type it needs. Conversely, the phosphate group and its attachments form a hydrophilic portion, which we call the hydrophilic head. And this means that they're water-loving. Remember, phosphate groups, which is one of the 7 important functional groups that we learned in the last chapter, the phosphate group contains a 2- charge, which makes it polar and therefore hydrophilic. We say that the phospholipid has a polar head and a non-polar tail. When phospholipids are added to water, they actually self-assemble into double-layered sheets called bilayers. This happens because water seeks to hydrogen bond with itself and other polar entities, which will expel any non-polar entities from the area. In this way, phospholipids will rearrange themselves so that the polar heads are on the outsides of the phospholipid bilayer in contact with water, and the hydrophobic tails orient themselves inward toward the interior. This forms a highly effective boundary between the cell, the interior of the cell, and its external environment. And we'll learn more about this in the next couple chapters. The third type of lipid is called a steroid. And you've no doubt heard of a steroid before, possibly used as a general term for someone who was ill, who needed to be on quote-unquote steroids. You'll see that steroids are actually an entire group of lipids characterized by this carbon skeleton consisting of 4 fused rings. An example is cholesterol, which is often talked about for its presence in our blood. High cholesterol levels are believed to contribute to cardiovascular disease. Cholesterol is one type of steroid. It is a component in animal membranes and a precursor for other types of steroids that we can make in our body. For example, estrogen and testosterone. Again, notice that 4-ringed structure in all of these molecules. That's what makes it a steroid. The third type of macromolecule that we're going to cover is proteins. Proteins are molecules with a wide range of functions and a vast diversity of structures. If you were to remove all of the water from a human body and analyze the remaining matter that we have, you'd find that proteins account for more than 50% of our remaining dry mass. Obviously, proteins are crucial to our life functioning. Let's note a few of the most important roles for proteins that they play in our biology. Enzymatic proteins. Nearly all biochemical reactions of our body require an enzyme. An enzyme's job is to act as a catalyst to speed up chemical reactions. We'll learn more about this in Chapter 8. Defense proteins, like antibodies, recognize foreign invaders such as viruses and bacteria, which is a crucial component of our immune defenses. Storage proteins, such as casein, which is a protein in milk, are important sources of amino acids for baby animals. Transport proteins carry molecules across membranes or through the bloodstream. Hemoglobin in the blood, for example, carries oxygen from the lungs to all the other parts of our body for cellular respiration. Hormonal proteins such as insulin are secreted into the bloodstream to coordinate an organism's activities. Hormones are a crucial part of maintaining homeostasis, for example, in regulating blood glucose levels. Receptor proteins are components of all cell membranes. It's their job to detect different signal molecules and respond appropriately. For example, this nerve cell is signaling this muscle cell to contract for movement. Contractile and motor proteins are responsible for movement, such as muscle contraction. And structural proteins are components of the cell that give support. For example, collagen and elastin are proteins that are found in our connective tissue, such as skin, which provides strength and elasticity. All proteins are made from this set of 20 amino acids. So amino acids are the monomers of proteins. When we make a protein polymer, it has a special name, which we call a polypeptide. Polypeptides are unbranched polymers built from amino acids. The term protein specifically means a biologically functional molecule that consists of one or more polypeptides. The 20 amino acid monomers are organic molecules with an amino and a carboxyl group. They all have this in common in their structure, and you can see that pictured here. The pink carbon that we see is called the alpha carbon, and then to the left of the alpha carbon, we have the amino group, which acts like a base, and to the right of that carbon, we have an acidic carboxyl group. So again, these are called amino acids. So we have an amino group and the carboxylic acid group. Carbon always needs, again, 4 bonds. So the third bond coming off of the alpha carbon is a hydrogen, and the fourth bond is a side chain, which we call R. The R group is the portion of the 20 amino acids that differ, right? So all of them have the bottom portion in common, and then they'll all differ based on what is present in that R group. The side chains are color-coded here. Yellow is nonpolar and hydrophobic. Green is polar and hydrophilic. Pink is acidic, and blue is basic. Amino acids differ in their properties due to their differing side chains, which are all the colors here other than purple, and those are all the R groups. So now that you are familiar with the monomers that make up proteins, our 20 amino acids, let's focus on how to build the polymers for proteins, which we call polypeptides. A polypeptide is a polymer of amino acids that can range from a few in a row to more than 1,000 connected. To build the polymer chain, the amino acids are linked by covalent bonds, and for proteins, the covalent bonds here are called peptide bonds. Take a look at the image provided here. We are shown 3 amino acids. Notice the portion of the amino acid monomers in the purple boxes are all the same. The amino group is connected to the alpha carbon, which is connected to the carboxyl group. The R groups here are shown in yellow and green. And as we build larger polypeptides, you'll notice that each has a unique linear sequence of amino acids, always with an amino or N terminus on one end, and the carboxyl or C terminus on the other end. Elongating the polypeptide bond involves, again, a dehydration reaction, whereby we lose a water molecule, just like when building the carbohydrate polysaccharides. Recall from Chapter 1 the concept of structure-function relationship. The specific activities of proteins are a direct result of their intricate 3D architecture. You just learned how the polypeptide is made by stringing amino acid monomers together with peptide bonds. However, creating a string of amino acids is not enough to have a functioning protein. Once we have our unique sequence of amino acids strung together, that polypeptide must go through an intricate process of folding before it can even be used in your body. A functional protein consists of one or more polypeptides precisely twisted, folded, and coiled into a unique shape. The unique sequence of the amino acids determines the protein's 3D structure, which again will determine how it functions. Once we have our properly folded protein, the function of a protein usually depends on its ability to recognize and bind to some other molecule. There are 4 levels of protein structure that you need to know, and they're termed primary, secondary, tertiary, and quaternary structures. The primary structure of a protein is the unique sequence of amino acids, again held together by peptide bonds. The secondary structure consists of coils or folds in that polypeptide chain. And the tertiary structure is determined by interactions among various side chains or R groups. And the quaternary structure results when a protein consists of more than one polypeptide chain put together. So we're going to look at these step by step. So again, the primary structure of a protein is its sequence of amino acids. The primary structure is like the order of letters in a long word. If any of these letters is misplaced or missing, the word will be incorrect and it won't serve its purpose. So the primary structure of a polypeptide is determined by the inherited genetic information that we have encoded in our DNA. In this image, you can see a polypeptide that is 127 amino acids in length. At the top is the amino or N terminus, while at the bottom we have the carboxyl or C terminus. As we move through the levels of structures, it's also very important to note what type of bond is involved at each level. So the primary structure here of a polypeptide is held together by the covalent bonds, again, which we call peptide bonds. The secondary structure of a protein occurs when we take our chain of amino acids, which is the primary structure, and we wrap it in a coil or we fold it. These two structures have special names called the alpha helix and beta pleated sheet. The image shows both of these secondary structures. On the left, we see the alpha helix. Notice the hydrogen bonds that are present between the very electronegative oxygen or nitrogen atoms and the partially positive hydrogens. Below, we see the beta pleated sheets. They seem to zigzag and run in opposite directions. You can see the arrow pointing right, then left, then right. Here too, we can see hydrogen bonds present holding these structures together. So make sure that you note in the secondary structure, there are covalent peptide bonds present, still holding the primary structure together, and now we have hydrogen bonds which are holding the alpha helix and beta pleated sheets in their structure. The tertiary structure of a protein consists of the overall shape of the polypeptide after it has been fully folded. This structure is held together by a bunch of different interactions between the amino acid R groups, and this is in comparison or opposed to interactions between the backbone constituents. The interactions shown here are hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals forces. In addition, we also are introduced to another type of covalent bond called a disulfide bridge, which exists between the sulfur atoms that are present in the amino acid cysteine. Recall from chapter 4, when we went through the functional groups, there was one called sulfhydryl, and these are called thiols. It was noted that these may be used to create disulfide bridges, so here we go, here they are. Again, these are covalent bonds, which are much sturdier than any of the other ones present, including hydrogen or ionic or hydrophobic. So those weaker bonds are able to break more easily under varying temperature or pH or salt conditions. Covalent bonds, on the other hand, don't break as easily, and these covalent disulfide bridges are very important in maintaining the protein's three-dimensional structure in our varying physiological conditions so that proteins can continue to function. Because if our proteins stop functioning, we stop functioning. And finally, the quaternary structure of a protein results when two or more polypeptides come together to form a larger macromolecule. So below we have two examples. Collagen, which you've heard before. Collagen is a fibrous protein credited with maintaining strength in our skin. It consists of 3 polypeptides, and we see them shown here in the different colors. We have red and green and blue, and these 3 polypeptides are coiled together like a rope. So what secondary structure are these 3 polypeptides individually? So remember, we have 2 different types of secondary structures. There are either alpha helix or beta pleated sheets. All 3 of these are in the alpha helix secondary structure. Hemoglobin is the protein in your red blood cells that transports and delivers oxygen throughout our body. It consists of 4 polypeptides, 2 alpha subunits and 2 beta subunits held together. You can see the heme groups present on these hemoglobins, each containing the heme iron responsible for its oxygen binding affinity. So we know that each protein begins with a unique and specific sequence of amino acids that are strung together by peptide bonds, which are covalent bonds. This specific amino acid sequence is inherited in our genetic code, which is the DNA. But what happens if there is a change in the primary structure? Even a slight change in amino acid sequence can affect a protein's structure and therefore its ability to function. For example, sickle cell disease is an inherited blood disorder that results from a single amino acid substitution in the protein hemoglobin that we just saw in the last slide. In this table, you can see the normal primary structure of hemoglobin and the mutated sickle cell sequence, which is only off by 1 amino acid, which is pictured here in red for you. The abnormal form of hemoglobin molecules become sticky and aggregate into chains, which causes the red blood cells to deform into a sickle shape, hence the name sickle cell disease. An interesting fact about sickle cell is that it actually protects against malaria, malaria is caused by a parasite that's passed from mosquitoes. The parasite relies on somebody having a healthy red blood cell as part of its life cycle. So the sickle cells prevent the parasite from thriving in your body. Sickle cell is a heritable disease, and if you receive 1 copy of the sickle cell trait, it's protective against malaria. However, the survival rate of individuals who receive 2 sickle cell copies is low due to that hindered ability to transport oxygen through your body. Of course, we really need oxygen as part of our cellular respiration process so that we can break down glucose to make energy. So there you go. You've now learned the 4 levels of protein structure, which lends to the 3D shape of a protein. In addition to primary structure, physical and chemical conditions can also affect the protein's 3D structure. Specifically, pH, salt concentration, and temperature all have the ability to unravel a folded protein. This process, whereby the protein's native structure is lost, has a special term called denaturation. A denatured protein is biologically inactive, meaning it doesn't have its shape, so it doesn't have its function. The body has many mechanisms for handling denatured proteins. In general, it will either attempt to refold it or it'll mark it to be degraded. One structure that's involved in protein folding in our cells is called chaperonin, and a chaperonin consists of a cylinder and a little cap on top of it. The unfolded polypeptide, so remember this is just that primary structure, it's just a chain of amino acids. So the unfolded polypeptide enters the cylinder from one end, and this causes the cap to attach. Once this happens, this signals the cylinder to change its shape, and this promotes a favorable environment for the protein to be folded in there. And then finally, the cap is released and the properly folded protein emerges. Diseases such as Alzheimer's, Parkinson's, and mad cow diseases are associated with misfolded proteins. And a cell that has too much denaturation of proteins will activate a process of programmed cell death, which we call apoptosis. And for our last macromolecule, we have nucleic acids. Students always forget this one, so make sure you note it down. Nucleic acids are the molecules that store, transmit, and help express hereditary information. Specifically, nucleic acids are DNA and RNA. DNA means deoxyribonucleic acid, and RNA means ribonucleic acid. Both are crucial components in the making of a protein. This process we covered in the first chapter a little bit is called gene expression, and we're going to cover it in depth in the 17th chapter. As mentioned in chapter 1, the amino acid sequence of a polypeptide is programmed by a unit of inheritance, which we called a gene. Genes consist of DNA, the nucleic acid made of monomers called nucleotides. Each gene along a DNA molecule directs synthesis of a messenger RNA, which we call mRNA, and the mRNA molecule interacts with the cell's protein-making machinery to direct production of a polypeptide. So in this way, DNA directs synthesis of mRNA and controls protein production. This flow of genetic information can be summarized as DNA goes to RNA goes to protein. And again, this is the process of gene expression. Let's break this down to the basics and take a look at the components of nucleic acids. To reiterate, there are two types of nucleic acids, DNA and RNA. Nucleic acid polymers are called polynucleotides. Image A shows a generic polynucleotide strand. Each polynucleotide is made of monomers that we call nucleotides. Image B shows the nucleotide monomer. Each of these nucleotides consists of three things: a nitrogenous base, a pentose sugar, and one or more phosphate groups. Image C shows all of the building blocks that can be used to construct a nucleotide monomer. Let's walk through building one together. We'll start with the pentose sugar. Remember from chapter 4 that a pentose is a 5-carbon sugar. We have two available here, deoxyribose or ribose. If you want to build a DNA nucleotide, you have to use the sugar deoxyribose. And conversely, if you want to build an RNA nucleotide, you have to use the sugar ribose. So next is the nitrogenous base. It's called nitrogenous base because these rings contain nitrogen. Notice that there were no nitrogen atoms in carbohydrates or lipids, but we did see them in proteins, and now we see them in these nitrogenous bases of nucleic acids. We have two types of nitrogenous bases. a single 6-membered ring, which we call a pyrimidine, and a double-ringed purine. The pyrimidines are C, T, and U, which are the abbreviations for cytosine, thymine, and uracil. And note that uracil is only found in RNA, while thymine is only found in DNA. The purines are A, which is adenine, and G, which is guanine. And then the final component is the phosphate group. And recall that phosphates are one of those functional groups that we learned about in Chapter 4. We also see these on ATP, for example. So if I want to build a thymine nucleotide monomer for use in DNA, I will need a phosphate group. The sugar that I'm going to need is the deoxyribose sugar, since I'm building DNA. And the nitrogenous base, I said that I'm making a thymine nucleotide, so I'm going to take the nitrogenous base thiamine. If we want to start building longer polymers of polynucleotides, like say we wanted to start building a DNA strand, nucleotide monomers are linked together by a new type of bond, a special bond called a phosphodiester linkage. A phosphodiester linkage consists of a phosphate group that links the sugars of two nucleotides. These links create a backbone of sugar phosphate units with nitrogenous bases coming off as appendages into the center of the double-stranded DNA molecule. And just to reiterate, the phosphodiester linkage again is a covalent bond. There are a few notable differences between the two polynucleotides, DNA and RNA, that you should write down. And I would suggest making a table for this. So the first is the structure of the DNA and RNA molecules. DNA molecules have two strands spiraling around an imaginary axis, forming that famous double helix. The backbones are running in opposite directions, which we call 5' to 3', and this is called running anti-parallel. One DNA molecule can encode up to 1,000 genes. RNA, in contrast, is a single-stranded molecule. The next difference is the type of nitrogenous bases that are used. We were introduced to 5 different ones. In DNA, A always binds with T and G always binds with C. And this is called complementary base pairing. Complementary pairing also occurs between RNA molecules. In RNA, T is replaced by U. So U is going to be pairing with A. And C still pairs with G here. This complementary base pairing is what makes it possible to generate 2 identical copies of each DNA molecule when our cells go through cellular division. And we'll learn about that in Chapter 12 and 13. In this slide, we can see all of those differences that I was just pointing out. So while DNA exists as a double helix, RNA molecules are more variable in form. Image A shows the DNA double helix. Notice that the strands are running in opposite direction. That's anti-parallel. So we have a 5' to 3' and a 3' to 5'. We also see the C nitrogenous base pairing with the G nitrogenous base and A pairing with T. The bonds that hold complementary strands of DNA together are hydrogen bonds, and you can see them as those dotted lines there. In image B, we see a special type of RNA called a transfer RNA or tRNA. This is a single strand of RNA that's just folded up on itself due to complementary base pairs within itself. Notice that G is still pairing with C, and that now A is pairing with U instead of T, because again, this is RNA, not DNA. And again, the bonds that hold the complementary base pairs here are also hydrogen bonds. And we'll see more on tRNA in the translation chapter. Once scientists uncovered the relationship between the structure of DNA and the amino acid sequence of proteins, biologists sought to decode genes by learning their base sequences. The first techniques for DNA sequencing were developed in the 1970s and have been refined over the last 50 years. In 2007, the Human Genome Project completed the first full sequence of the human genome, a passion project that took nearly 20 years. Since then, science and technology have synergized for rapid development of faster and much less expensive sequencing models. To date, thousands of genomes have been sequenced, generating large sets of data, and you can find any of these genomes through a website called NCBI. Bioinformatics uses computer software and other computational tools to deal with the genome sequencing data of all of these organisms. Analyzing large sets of genes or even comparing whole genomes of different species is a science called genomics. A similar analysis of large sets of proteins, including their sequences, is called proteomics. And that will wrap it up for this chapter. As I mentioned at the beginning of this video, this chapter is a full chock-a-block of nuanced information about the 4 macromolecules. So if you haven't yet, take a few minutes to organize your notes into a table for better retention of the information. In the next chapter, we're going to be talking all about the cell.